Thank you, everybody, for coming. I'd like to go ahead and get started so we can keep our session on time. My name is Melanie Cree-Green. I'm from the University of Colorado, and Elizabeth Parks will be joining us shortly. And we're the moderators for this session of new methods and models to study human metabolism with stable isotope tracers. And just to let people know, this is a AFMR-sponsored symposia here. And the mission of AFMR is to develop and mentor tomorrow's leaders in medical research, from bench to bedside to practice. And just as a little bit of history, AFMR was actually founded in 1940 by Dr. Henry Christensen, and it has consistently been an organization that has offered young researchers the opportunity to present their findings to peers and also to receive the guidance of senior scientists. And the approach to AFMR-based research is multidisciplinary and translational. And in 1996, it was renamed to AFMR and has been associated with FASEB for more than 20 years. And so currently, AFMR has 4 regional meetings that are held between January and April. These are great opportunities to have students present their work. They have many oral sessions for students. And then AFMR participates in 2 national meetings, Translational Science and then here at FASEB. AFMR has 2 publications, one of them the Journal of Investigational Medicine. And of note, today's symposia will be published in JIM later this year. So you can look out for that. And then the Journal of Investigation High Impact Case Reports. And AFMR has been very involved in public policy. The 2 kind of biggest achievements for AFMR were significant roles in getting the Mentored Clinical Scientist Research Development Awards, K08s and K23s, program funded, and then also with the Clinical Research Enhancement Act for NIH loan repayment program. And currently, AFMR is aligned with Research America and FASEB for continued public policy efforts to fund research. So a little bit about background of stable isotope tracers at FASEB. Stable isotope tracers have been presented at FASEB since at least the 1960s. And almost every year there has been a consistent tracer focus, ranging from symposia like this, evening industry-sponsored symposia, to separate meeting tracts. But despite this 60-year history of this methodology, we still have so many questions in physiology that are still to be posed. And so the field continues to move forward in terms of needing new methodologies and new ways to interpret the data that we're collecting. And so with that, we're going to have a very diverse group of talks today with different ways of using stable isotopes for different physiologic measures. So the first talk will be Advances in Isotope Models for Understanding Protein Metabolism, by Dr. Robert Wolf from UAMS, and you may recognize his name from the Stable Isotope Tracer textbook. Our second talk will be use of an oral glycerol drink combined with isotope analysis to understand hepatic metabolism. This talk is going to be discussing use of magnets for isotopomers rather than the traditional LCM-SMS. And then our last talk will be adapting oral glucose tolerance tests with tracers to different patient populations. And here we're going to delve a little bit into mathematical modeling, which is also required to help us understand some more complex tracer challenges. And if you could please, we'll have a couple questions at the end of each session, at the end of each speaker for about 5 minutes, and then we can do more questions at the end of the session. And so with that, I will have Dr. Robert Wolf come up. See this one showing this. You think? Gotcha. Gotcha. Okay. Go away. Go away. Go away.
Okay, kind of a shaky start, but let's hope things pick up from here. I think we want to, okay. Usually a perfunctory thanks for being included, but when I heard in the introduction that this AFCR is particularly to promote the development of young investigators, I really am happy to be included in the group of speakers. I changed the title a little bit because I actually didn't have the title of the symposium at hand when I prepared the talk, and as you'll see, we're not really going to focus in this talk on a new method. In fact, whole body protein turnover, which is the main topic, this study I was able to find from 1939. So it's certainly a liberal interpretation of new methods to say I'm going to focus on this whole body protein turnover. But I think it's an interesting circumstance for a few reasons. The most important being, if you're doing this kind of methodology, that there are considerations, although the methods have been around for 70 years, that have not really been appreciated. And I think that there's been a new interest, particularly as we talk about translating research into clinically relevant approaches, that the whole body protein turnover studies still really have a place. Because for one thing, we know when we're talking about the measuring the response to nutrient intake, that it's at the whole body level. The whole body eats it, so looking at the whole body protein turnover has relevance. And there's certainly been much more of a focus on muscle than any other aspect, but muscle constitutes less than half of whole body protein metabolism. So when we focus just on the muscle, we are missing things. And using the whole body protein approaches, of course, doesn't preclude use of this simultaneously of measurement of response in muscle. And that's the third point here. And the fourth point being that, and this is really, I think, becoming much more recently appreciated, that both protein synthesis and breakdown are measured, and that a lot of the anabolic responses to nutrient intake is the result of changes in protein breakdown rather than protein synthesis, which is, of course, missed by most traditional approaches. Well, the whole body protein, I'm going to use this because this mouse isn't working. I wonder why it's not moving. I thought we had gotten this straightened out, but you know, the thing is, there's no. Yeah, we got no.
We need to hit that.
Yeah, but where's the mouse? Okay, now it's working its way up. Okay, there we go. There it is.
There you go.
Okay, that should work. Okay. So here's the slide I was talking about when I said that there's nothing new about whole body protein turnover, and the points I made about why we're still interested in a method that's been around for so long. Well, the model that I'm going to talk about, and that's really most widely used, involves use of essential amino acids as a tracer. And the point is, in the fasting state, since essential amino acids and leucine and phenylalanine would be the most commonly used example, but it could be done with any essential amino acid, the only source of amino acids coming into, of that amino acid coming into the amino acid pool is from protein breakdown. So by the traditional tracer dilution method, we're able to quantify the rate of protein breakdown. If we also are able to measure its oxidation, in the case of leucine, it's actually oxidation. In the case of phenylalanine, we use the rate of hydroxylation to tyrosine. But irreversible loss of amino acids, then the rate of synthesis can be calculated as the rate of breakdown minus the rate of oxidation. And this works out just fine in the fasted state. The problem becomes a little more complicated when we get into the postprandial state, which as we start to really look at how nutrients are handled in the body, obviously we need to focus on the postprandial state, in which case now the tracer measures total appearance of amino acids. That is not only coming from breakdown, but also from exogenous RA. The amount of, let's say if we're using leucine as a tracer, the amount of leucine that's coming into the amino acid pool from the protein, dietary protein, in addition to this, so that we have to be able to quantify how much the exogenous contribution is to the total RA, so that we can calculate endogenous RA, which is equal to breakdown, and then from that we can calculate the rate of synthesis. Well, there have been different approaches, but certainly the most elegant and simplest approach, it's called the intrinsically labeled protein model, which was first described in the 90s, but has been used many times in a variety of formats since then. The general idea is that we label the exogenous protein or the dietary protein with the same amino acid that you're using as the tracer, but a different label on it, so that we can quantify through the tracer appearance in the peripheral pool what the actual absorption or contribution of the exogenous RA is to the total RA. The protein can be produced by, for example, feeding labeled amino acids to cows and using the milk that's produced from the cow that's labeled intrinsically with the protein, or growing chickens on a C13 amino acid substrate and isolating the eggs. So a variety of approaches, but in fact this has become considered the gold standard. In fact, what motivated me to start looking at this method in a little more detail was the fact that we had a paper recently rejected because we didn't use this labeled endogenous approach. So that was tangible sort of motivation to take a closer look at this intrinsically labeled protein approach and see if it really is as simple and elegant as it would seem from this schematic here. Once the, I'm just going to show this real briefly, and there are a few things I'm going to skip over, but just to point out that to calculate the total RA, we use the Steele equation, which again, nothing new, it's described in the 50s. But the important point is that, the way that intrinsically labeled protein pool is quantified in, turnover is quantified in the case of the exogenous RA, is that you actually use this component, which is the total RA times the enrichment in the blood coming from the PO, or the ingested protein. So that instead of actually having a traditional application of the Steele equation, the appearance of label is actually an unknown in the equation. And we'll talk a little bit about this specifically, the problems that brings up in a little bit. But first I'd like to just go through some of the major assumptions. And those of you that aren't doing protein metabolism, please try to just think in broader terms, because these same issues, I can tell you, come up over and over in a variety of tracer methods. And so that you really need to think about the ways in which the tracer can give you an erroneous result, because to sort of spoil the end point here, that's the answer here is that the intrinsically labeled protein, in fact, doesn't give you a good measure of the exogenous RA. Well, why not? That means that some of the major assumptions, some or all of the major assumptions must be wrong. What are the assumptions? First, that when you ingest a labeled protein, that what's absorbed is actually the enrichment that you ate and were able to measure. But in fact, we have to assume then that there's no dilution of the ingested label prior to the measurement of the peripheral blood. Secondly, we have to consider the measurement of the label from intrinsically labeled protein in the non-steady state, and that there's no recycling of tracer, and that the volume of distribution can be reasonably approximated by a single value. So I'm not going to go into detail on all of these points. Some of them have been really hashed over before. But I am going to point out, start with this issue of the dilution of the intrinsically labeled protein in the GI tract. Because the GI tract is continually losing protein and amino acids. And in particular, when you eat a protein meal, then there's digestive enzymes that are secreted into the intestines, and those protein are also digested, those amino acids become contributing to the total amount of amino acids that are absorbed. And of course, they're not labeled, they're coming from endogenous proteins. So what we I'm going to skip over the derivative of where these numbers come from, but we know from the model that's published in that paper there that this unlabeled source of amino acids into the GI tract dilutes the labeled amino acid that's ingested in the intrinsically labeled protein. So if we just look at specific numbers, and now I'm not going to go through all this derivation, but just take my word for it from the papers that we calculate that this is the rate of appearance of leucine, the rate of absorption of leucine from the digestive of endogenous proteins. So these are unlabeled amino acids. If we look at 20 grams of whey protein ingested and we account for the digestibility, we end up with a total of 0.0, and that the enrichment is 0.05 tracer-tracy ratio, then we have an appearance of a total appearance of this amount. This amount is the amount of labeled leucine in the protein, and this is the amount of labeled leucine in the unlabeled leucine. So if we put it together, what does all this mean? Well, what it means, if we take average values for secretion of protein into the GIT and consider that in the context of a 20-gram dose, it's a pretty big amount. So that the total amount of protein that of leucine that we're getting is 2.1 grams. Out of that, a significant amount, 0.6 grams, is the unlabeled endogenous. So what does that do to our enrichment? The labeled isn't affected by the absorption of the endogenous, so that we look at the tracer-tracy ratio and it's 0.038. We started with a value of 0.05, so it's a 24% dilution. Of course, this is an approximation. We don't know exactly what it is, but the basic point is that we know that the magnitude of secretion of unlabeled amino acid into the GIT is in the same ballpark as the amount of dietary protein that's eaten. So we know from this that the tracer intrinsically labeled protein is diluted in the GIT. And what this means is, if you remember, the reason I showed those equations is just to highlight the key point of why does it make a difference. And that is that the presumed enrichment of the protein in the calculation of exogenous RA is the measured value at the point at which you eat it. But what we're seeing is that the actual value that's absorbed is not 0.05, but a 24% dilution of that. So what this means is that if we take that in the context of how do we calculate endogenous RA, which is equivalent to protein breakdown, that an underestimation of the exogenous RA from the intrinsically labeled protein approach will cause an overestimation of the rate of breakdown. This is not encouraging. Well, this will help get me back on time. The bottom line is, so what does this mean? And the data that the blank slides there represented calculating the rate of exogenous RA with the assumed value of 0.05 versus the accounting for the tracer dilution, and showing that you have different rates of protein breakdown calculated. And in fact, what it shows from the results is that if you eat 20 grams of protein and use the intrinsically labeled protein method, you conclude that there is no suppression of protein breakdown. If you account for the dilution, then in fact you get a significant rate of suppression of the rate of protein breakdown. If we look at other approaches where we've looked at both whole body as well as across the forearm and measured with an independent method whether essential amino acids actually suppress protein breakdown, we see that a variety of studies show that in fact the intake of essential amino acids has a suppressive effect on whole body as well as muscle protein breakdown, so that the conclusion using the intrinsically labeled protein method is physiologically incorrect, that it concludes that that the rate of protein breakdown doesn't change, therefore it's not really playing a role in the anabolic response to the dietary protein, where in fact it's playing a major role. And in fact, what we find is really a larger role than the stimulation of protein synthesis. Now, another potential area where the tracer can be diluted is across the splanchnic bed. And this is a problem that comes up over and over in tracer studies where incorporation is measured. So it really has a application far beyond this particular example. The basic point is we'll assume a situation, obviously this is just a schematic, but a situation where protein synthesis and protein breakdown are going on continuously in the liver, but there's no net difference between the two. So the amount of amino acids taken up by the protein is equal to the amount that are released. However, if we consider, let's say, an enrichment, a tracer-tracer ratio of 0.25, meaning that 1 out of every 5 of the molecules of tracer that enters the portal vein here is labeled, the uptake of label into the liver is going to be proportional to what it is in the blood coming in. But what leaves the protein pool is going to be reflective of what the protein pool is. And when we first start the tracer intake, there's no label here, so that you're going to have uptake of label, but you're going to have no release of label, with the result that there's no net change in the amount of tracer or tracee that's come in, and there's no difference in the amount of tracee from what's come into the liver and what's leaving, and yet you've had a 33% reduction in the actual amount of tracer that's leaving, and therefore another dilution of the tracer enrichment. Now, of course, over time, the protein pool becomes more and more labeled, and this will ultimately, at a plateau value, what comes out will equal what goes in, so that we can't give you a precise amount of how much of dilution occurs this way, but it's certainly a significant site of contribution as well. We know then from this that, in a general sense, protein turnover in the splanchnic tissue and organs can dilute the enrichment of the intrinsically labeled protein, and again, it's the same as with the dilution from the gut, and that is that if it's diluted, it will overestimate the rate of protein breakdown and be contributor why we have had such a number of papers that have concluded that amino acids and protein don't suppress protein breakdown, which are really just a methodological the result of the methodology rather than the physiology. Now, the final point I want to hit on is the issue of how it's calculated, how the appearance is calculated. Because, of course, when you eat it, even eating as a bolus, that comes in over a period of a couple of hours so that there's a non-steady state. And as I said, the equation, the Steele equation, is rearranged to actually measure the rate of appearance of the label as the unknown, rather than in the case where the Steele equation was actually derived, the tracer infusion is the known variable. In this case, it's the unknown variable. And we have to assume, in this case, that there's no recycling, so that whatever we measure in the blood, it's just the result of what's been absorbed, not that it's been recycled around the body. But we know that that's not true, that it's not possible. The point is here that this is the term that we're measuring, namely the appearance of the isotope coming from the intrinsically labeled protein, and it's a variable over time. So we just are taking, if we even take the point of taking the total area under the curve, we still are overestimating what that appearance is because of the fact that there is recycling of the tracer. And the most obvious example of that is the injection, the response to the injection in plasma leucine of labeled and unlabeled leucine together, which is used in the flooding dose technique. And what we see is that the appearance is a bolus, intravenous, and it occurs before this first sample is taken. And yet, even 2 hours after that appearance has stopped, because the infusion is just a bolus, there's no more coming in, and yet we still have enrichment in the plasma, 50% of what we started with 2 hours later. And the reason is because there's recycling of the label. So that as we try to measure or quantify the rate of appearance of the intrinsically labeled protein, then it's recycling what you're measuring at any point in time is what's just been absorbed as well as what's recirculated that hasn't been cleared completely in the previous pass of the tracee across the active tissues. So another source of error, and all three of these sources of errors, the important thing to recognize is this isn't just random effects, that in fact this error of recirculation works in the opposite direction of the other two, and that's why you get kind of ballpark reasonable answers with the methodology, because you have certain assumptions, namely related to the dilution of the tracer, that cause an overestimation of an underestimation of the exogenous RA, overestimation of the rate of breakdown. In this case, we're overestimating the appearance of tracer because we're double counting molecules as they've first appeared and then recirculated, and then as a result we're overestimating the exogenous RA and underestimating the endogenous. So we have three major sources of errors. One tends to cancel out the other two, but that really doesn't help in terms of valid conclusions. So what's the alternative? And you know, as Melanie was referring to the tracer history of the EB, and for at least 15 years I was the organizer of the tracer methodology group. So going back to the 1970s, I have always loved tracers, but the thing that's interesting is sometimes it's just not the best way to skin the cat, because what we find is the alternative to the endogenous, the intrinsically labeled protein, we can call the bioavailability approach, which is much more simple, but we know the amount of protein we've eaten, and we know the true ill digestibility. Almost all the proteins have that's been experimentally determined, and it's ranged. What this refers to is how much of the protein you've eaten, the amino acid tracee in the protein you've eaten, is actually absorbed in the form that you've eaten it in. And for high quality proteins, that's not very variable. It's in the range of 90 to 100%, but these values are available. And then in the case of the, but what you still have to account for is the fact that when you eat it and it passes through the splanchnic bed, there's still some of that tracee is going to be cleared by the splanchnic bed before it ever gets to the periphery. But we know in the case of phenylalanine exactly what that is, because phenylalanine is only cleared in the body and the liver. So that we measure the rate of phenylalanine hydroxylation. This is in fact the fraction of amount of phenylalanine passing the liver that's taken up and irreversibly hydroxylated, meaning that it's a direct measure of the total clearance. We don't have to worry about protein turnover, because if there's no net difference between synthesis and breakdown, there's not going to be any net uptake of the change in bioavailability, where it is a big problem in terms of the intrinsically labeled. The same thing with your bioavailability of the ingested amino acid. It's diluted by unlabeled, but we're measuring unlabeled anyway, so it doesn't have an impact as well. So that it's a circumstance where really that due to the inherent problems of labeling, of using an intrinsically labeled protein, that we're actually much better off and much more accurately off by using just the calculated bioavailability. Parenthetically, which was what my point was in the paper that was rejected, and it goes to show that when you reject a paper based on your favorite method, you have to prepare for the fact that the person who gets rejected may look at that method a little more closely. So you live and learn. Anyway, to wrap things up, I'll just summarize by saying that this intrinsically labeled protein is well considered to be the gold standard. In fact, the fundamental assumptions underlying its use are pretty much all violated. The intrinsically labeled protein is diluted in the GIT and across the splanchnic bed, resulting in overestimation of exogenous RA. The overestimation of exogenous RA underestimates the suppression of protein breakdown, so that we've had really some incorrect concepts for the last 20 years as to how dietary protein modulates the anabolic response in humans. And thirdly, that this recycling of tracer may counter the effect of the dilution of tracer to some extent, but it's unknown how much that is, so that really it's, you come up with somewhat reasonable values, but only because of canceling errors. And so the final, by comparison, the use of the bioavailability approach is we have a direct measure of the only real source of loss of the phenylalanine. And as a result, of course, there's always error in measurement, but the error isn't systematic. There's no over or underestimation. The error is just a random error related to measurement. So from all this, I conclude that the exogenous RA from ingested protein is better estimated by the bioavailability approach than by the intrinsically labeled protein approach. I don't know if I made up for our shaky start on time, but that's the end of my talk. Thank you.
We can take a couple questions if you'd like to come up to the microphone, please.
I can tell there's probably nobody in the audience that's used this method because yeah, I was definitely prepared for whatever attack I could receive.
So would you suggest people go back and recalculate their data with this alternative approach to
Yeah,
see?
I mean, I think we've actually, there are a few of the papers really, and in fairness to the originators of this method, they provided all the data in their paper to recalculate, which we've done. And as I said, it gives you complete, I mean, it's a little bit hard because we don't know the magnitude of the errors, but the fundamental, there are a couple of fundamental issues that have come from this, that when the recalculation, one is that the speed of digestion affects the rate of protein synthesis. And that's also, I haven't gone into that, but that's also a result of this when you recalculate the data, making approximations for the assumptions, that you don't get that conclusion. And the second, the most important being that no matter how you do it with this method, you conclude that protein breakdown doesn't play a role. But when you recalculate it with the bioavailability approach and all of their other data, then it shows just a significant drop in protein breakdown. So it's really a fundamental basis for, if we think about how much research has been done on protein synthesis and how little has been done on breakdown, and yet when this method is used, you find that the magnitude of effect of protein breakdown far exceeds the effect of protein synthesis.
How do you deal with a lot of the amino acids coming in as peptides into the circulation?
Well, they're not, that's why the term true ileodigestibility. So that the question is, how do you deal with peptides? And if they're coming in as peptides, they're not coming in the Tracy format. So they are excluded. That's part of the equation for the calculation of true ileodigestibility, that you exclude anything that's not in the format. Now, of course, they could ultimately be converted into amino acids in the body. So, that would be a potential source. But if you look at you you probably could account for it if you had in the animal experiments, the way they did the true ileodigestibility is taking samples from the end of the ileum. And so you might be able to make assumptions there, but the way the method is worked here, you're only accounting for amino acids that are absorbed in the pure form.
All right, thank you.
Our next talk is by Yoon-Suk Jin. It's entitled Use of an Oral Glycerol Drink Combined with Isotopomer Analysis to Understand Hepatic Metabolism.
All right, thank you for this opportunity to present my data here. And today I'm going to talk about carbon-13 labeled glycerol to study liver metabolism. And the liver is the major gluconeogenic organs in our body. When our blood glucose level declines under a fast, the liver make a new glucose through gluconeogenesis. And there are many gluconeogenic substrates. The major one is lactate and pyruvate and some amino acids, also gluconeogenic substrate. Glycerol is also the gluconeogenic substrate. When triglyceride, the fat in our body hydrolyzes, it produces fatty acids and glycerol. The glycerol in our body are most utilized by the liver because the liver has very high content of glycerol kinase, becoming glycerol-3 phosphate. When this glycerol-3 phosphate converted to these two trioses, dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Anyway, these two are in very rapid exchange. The condensation of these two trioses make glucose. And I study liver metabolism using the stable isotopes and NMR. When liver is exposed the labeled glycerol, for example, the uniform labeled glycerol, I expected to see 1, 2, 3, and 4, 5, 6 labeled glucose from the liver. So when you look at the glucose using carbon-13 NMR, I expected to see this kind of spectrum. Here's the carbon-13 of glucose having 1, 2, 3, and 4, 5, 6 labeled glucose. It's precisely glucose derivative, but I just call it glucose. We are looking at glucose C2 and C5 positions. In both areas, you see the singlet. This is coming from glucose C2 labeled, carbon C2 labeled glucose, and carbon, the 5 position labeled the glucose. So in our body, we have a 1% natural abundance, carbon-13. So I'm not going to talk about the singlets that much, but if you see here in both areas, we can see quartets here. In C2 region, the quartet signal coming from 1, 2, 3 glucose. This is C2, the carbon coupled with the C1 and C3 at the same time, so it gives a quartet signal here. And similarly, you see quartet signal in C5 from the 4, 5, 6 labeled glucose. In this case, one peak from quartet is overlapped with the singlet, so that's why you see only 3 the obvious peaks from here. Anyway, this is what I expected if I think the liver is exposed with the labeled glycerol. And however, it looks different. The actual spectrum looks different from previous one. But we are not that bad. As you see here, C2, C5, we see large peaks from quartets in both positions coming from 1, 2, 3, and 4, 5, 6 glucose as we expected. However, we also see a bunch of doublets, 1, 2 labeled glucose, 2, 3, 4, 5, and 5, 6 labeled glucose. How come we have a double labeled glucose after treating the triple uniformly labeled glycerol? This can be explained by the labeled glycerol, the metabolism through the TCH cycle prior to gluconeogenesis. So labeled glycerol, become this trioses. When this triose, instead of going to the gluconeogenesis, it may undergo glycolysis, becoming labeled pyruvate. So far, the labeling pattern from glycerol to pyruvate, it does not change. They are all the trioses. However, when pyruvate enters the TCA cycle, the labeling pattern will be changed. Pyruvate enters the TCA cycle through carboxylation and becoming oxaloacetate. And also, it can enter the TCA cycle after losing 1 carbon through acetate coenzyme through pyruvate dehydrogenase. After entering the TCA cycle, the labeled TCA cycle intermediate, the labeling pattern will be extensively scrambled. And one of them, the intermediate of TCA cycle is oxaloacetate. This labeled oxaloacetate can exit the TCA cycle through PEPCK, phosphoenol pyruvate carboxykinase. And this PP triose, the major labeling pattern in this case is double labeled one after you're scrambling the carbon-13 labeling. And the 2,3 is the major one, a little bit 1,2 will be produced. When these trials, again, participate in gluconeogenesis, the 2,3 trials become 5,6 glucose or 1,2 glucose. In the case of 1,2 labeled 1, it will become 4,5 or 2,3 labeled glucose. So now we can explain how come we have double labeled glucose isotopes after treatment uniformly labeled glycerol. But here we still have one more question by looking at this. And actually, I was really shocked when I first noticed AA12 and AA56. They are so different in here. Because these two originated from the common source, I expected a similar peak size, similar the concentration of 1,2 and 5,6 glucose, but they were not the same. And after thinking the other pathways, I thought that the pentose phosphate pathway activity explains the difference. Here again, the glycerol direct incorporation to gluconeogenesis will be produced 1, 2, 3 or 4, 5, 6 labeled the hexose. If these two isotopes enter this pentose phosphate pathway, if you're going to use the labeled, in the case of this one, it's going to use the carbon 1. It was labeled. In this case, it's going to use the carbon 1, but this is not labeled. So they become 1, 2 labeled pentose or 3, 4, 5 labeled pentose. This pentose will be scrambled. The carbons will be rearranged through non-oxidative phase of pentose phosphate pathways, and it's going to produce again the hexose. In this case, 1, 2 glucose, precisely fructose 6 phosphate will be produced from 1, 2, 3, the hexose. Here, in the case of 4, 5, 6, the labeling pattern does not change. It will be the same labeling pattern. So when the gluconeogenesis interrupted by this pentose phosphate pathway, it's going to produce additional 1, 2 glucose. That's why this 1, 2 glucose and 5, 6 glucose, they are different. So by looking at this carbon-13 labeling in glucose after labeled glycerol treatment, we can get quite important information about the liver metabolism. Obviously, glycerol metabolism to make incorporation to glucose, and we can measure direct incorporation or indirect incorporation through the TCA cycle. The indirect contribution is important. It's because TCA cycle exists in mitochondria. So this double-labeled one informs the mitochondrial biosynthetic functions. Of course, using this difference, the pentose phosphate pathway also can be estimated. And this pathway produces NADPH and the pentose. These two major products are important. The pentose, of course, is used for the nucleotide synthesis. Also, NADPH is the reducing equivalent needed for the reductive biosynthesis, such as lipogenesis, the cholesterol synthesis, and it also used to make a reduced form of glutathione. Glutathione is it fights against the oxidative stress. Okay, that one is based on glucose analysis, but liver also make another important metabolite, the triglyceride. So here, the phosphorylated glycerol 3 phosphate, instead of going nucleogenesis, it may participate in the fatty acid esterification, producing triglyceride. So when you see the triple labeled glycerol backbone in triglyceride, it informs glycerol direct incorporation to triglyceride. Okay, we can also expect it to double label the glycerol backbones. So now it's easy to understand this one. So this double labeled one, again, the glycerol experienced the TCA cycle prior to incorporation to triglyceride. This is the carbon-13 NMR of triglyceride, focusing on glycerol backbones. The right one is a glycerol backbone C1 and C3 positions. Here, the doublet, this signal coming from a double-labeled and triple-labeled glycerol backbones. On the left one, it's a glycerol backbone C2. Here, In this reason, we can distinguish double-labeled one and triple-labeled one. Again, this doublet reflects glycerol metabolism through the TCA cycle, and triplet reflect the direct incorporation to glycerol backbone. Okay, so using this glycerol, we can get that kind of information about the liver metabolism. If you do this experiment using animal models, it will be no problem. You can freeze clamp the liver and do the lipid extraction or isolate the glucose and convert to the glucose for NMR analysis. However, if you apply this method to human study, it's not easy to have liver tissues from humans. But luckily, these two metabolites, glucose and triglyceride, synthesized in the liver, are released into the circulation. And so we can study the liver metabolism using the blood, triglyceride, and glucose. And with the animal models, I studied both liver samples and blood samples, and the information is basically the same. So this glycerol method I originally studied using animal models, and I tested with several different animal models. And the method developed in animal models, I translated to the clinical studies. And now I have a list here that clinical studies that I have been involved. Some of them are already published, and some of them are going on. Some of them are doing with a collaboration with the others. And here, Dr. Green, and she also applied the uniform labeled glycerol method to study liver metabolism of girls with polycystic ovarian syndrome. And actually, she combined the glycerol method and oral sugar tolerance test. And today, for the remaining time, I just briefly show you some results that my study with the fatty liver subject. And this one recently published. Okay, in this study, we recruited obese volunteers, and we measured their liver fat content using proton, the NMR of liver. Here you are looking at two spectra from two subjects. The left one is from lean, normal liver. This is a water signal, big signal you can see in both spectra. And here you see very small signal coming from triglyceride. And this person has a 1% liver fat, normal liver. The right spectrum, you see the huge peaks coming from triglyceride, and this person has a 14% liver fat. So based on this liver fat content, we divided two groups, normal groups and fatty liver group, who had more than 5% liver fat. And this table shows you the clinical and biochemical characteristics between two groups. BMI, age, gender, all these are the same. Even blood test result, there's no differences. However, the only difference is liver fat content. Normal group, 2% average liver fat. High fatty liver group, they have 11% liver fat. So these actually, patients with a fatty liver, but they are subclinical condition. This is a study protocols. All the volunteers, they were overnight fasted. On the study day at 9:30, they drank water, having the dissolved uniform labeled glycerol. After that, we collected the blood at 6 time points, beginning 30 minutes up to 4 hours. And actually, there's one more tracer in this particular study. Here, as you see, we also gave a DTO. Maybe you are familiar with the DTO method introduced by Dr. Landau. Using this DTO method, we can distinguish how many your blood glucose coming from glycogen or gluconeogenesis. Okay, so plasma sample prepared for lipid extraction, and glucose was derivatized for NMR analysis. Here's the first result based on triglyceride, glycerol backbone C1 and C3. The left one is from normal the liver, the right one is from fatty liver. As you see, his doublet, this signal coming from this labeled glycerol backbone is much low, more diluted. So when you see this graph in the middle bottom here, the open circle is from normal liver, the dark black circle is from the fatty liver. The enrichment is lower in fatty liver group. It's the percentage. When I calculated the absolute concentration of triglyceride with the extra, excess carbon-13 enrichment, the concentration is the same between two groups. So this means the fatty liver group, they diluted the tracer, diluted more because they have a larger triglyceride pool size in their body. Okay, this data is based on glycerol backbone C2 analysis. Here we are comparing the doublet and triplet signals. Doublet signal informing glycerol metabolism through the TCA cycle, triplet direct incorporation. When you compare the relative size, this doublet is much bigger in fatty liver group. So here, this graph shows the percentage of indirect contribution to glycerol backbone of triglyceride. Throughout the time points, the fatty liver group always higher the enrichment. This is the absolute concentration of triglyceride with double labeled triglyceride backbone. Not much differences, but trends are different. Especially if you see the Tmax, the time for maximum concentration is 125 minutes for fatty liver group and 190 minutes for normal liver group. So this graph tells you the more fraction of labeled glycerol experienced to the TCA cycle in fatty liver group, and this process is quicker in fatty liver group. Now the data based on glucose analysis, the carbon-13 enrichment in glucose is lower in fatty liver group at the beginning at 30 minutes and 60 minutes time points. This is the absolute concentration of carbon-13 labeled glucose, not statistically significant, but still there's a trend. At early time points, fatty liver group has a low concentration. Okay, 5-6 labeled glucose production. Informing glycerol metabolism through the TCA cycle prior to gluconeogenesis. There's no statistical significance, but when you see here, after 60 minutes, the average value is higher in fatty liver group. And actually, at the time point 120 minutes, this concentration is higher in fatty liver group. So the fatty liver group, more glycerol experience the TCA cycle before the gluconeogenesis. The ratio 5-6 over 4-5-6 in glucose informing indirect contribution through TCA cycle over direct contribution. Not statistically significant, but the average value is always higher in fatty liver group. The 1,2 glucose production through the only pentose phosphate pathway. Again, not significantly different, but there's a trend. Fatty liver group has more 1,2 glucose production. In animal model, the fatty liver animal actually had higher the pentose phosphate pathway activities, and they have a higher lipogenesis. So we can, based on this, maybe NADPH from the pentose phosphate pathway used for lipogenesis. Okay, the last NMR spectrum of my talk, and this one looked different. It's not anymore carbon-13 NMR. Now we are looking at the deuterium, the NMR. We are looking at deuterium attached to glucose. When liver produces glucose in the presence of D2O in your body, it's going to label as specific positions of glucose depending on its sources. When glycogen and gluconeogenesis both produce glucose, both of them will label H2 position right here. That's why the H2 peak is the biggest peak here. If the glucose coming from gluconeogenesis, it's going to label H5 here. In gluconeogenesis, if it's originated from the TCA cycle, not glycerol, from the TCA cycle, it's going to label only H6 position. So based on the peak area, H2, H5, and H6, we can calculate relative contributions from glycogen, glycerol, and the TCA cycle. And when I measured those contributions, at each time point, there's no differences at all between normal liver and fatty liver. This graph, left one is from normal liver, the right one is the fatty liver. However, when you see the two graphs, you see some differences here. From the normal liver, this is glycerol contribution, glycogen contribution, and TCA cycle contribution. After glycerol load, the contribution from glycerol is increasing right here. At the time point 60 minutes, it reaches the maximum. After that, it declines. The same thing happened in fatty liver group. However, the process is much slower in fatty liver group. Another thing that I thought interesting is the glycogen contribution. As a glycerol contribution increases, glycogen contribution decreases. As a glycerol contribution declines, glycogen contribution increases. It's the same trend right here in fatty liver group. However, the TCA cycle contribution did not change throughout the study period. So when glycerol contribution are changed, the glycogenolysis will respond accordingly. So the conclusion of this study is fatty liver patients, they have lower carbon-13 enrichment in the glycerol backbone of triglyceride. They also have a higher glycerol metabolism through the TCA cycle, and they had delayed gluconeogenesis from glycerol. Also, they are less flexible in adjusting, supporting fluxes of glucose production after an oral glycerol load. Okay. Dr. Molloy cannot make this meeting, but he is very supportive of all my studies. Rebecca prepared the samples, and here the bunch of nurses are involved in this study. And thank you for your attention.
This paper is now open for questions. I have one. So is there just more flux down glycolysis and gluconeogenesis in the patients with fatty liver that's increasing the dilution? It seems to be your conclusion.
It should have a glycolysis. Also, the TCA cycle must be more active in the fatty liver. And it is known the fatty liver, they have some people reported higher fluxes through the TCA cycle. I think they contributed higher contribution through the indirect pathways.
You didn't measure hepatic glucose production when you were doing this, right?
Not the absolute,
Yeah.
turnover. I didn't measure it.
Yeah. Thank you. Yeah, Bob.
Well, first I'd like to congratulate you on a wonderful paper. But one of the things I was wondering when you were presenting this was kind of the analogy to the schematic I showed with protein metabolism, where you have no net change in protein synthesis or breakdown, the balance between the two, but you have tracer uptake, which is gradually labeling that pool and unlabeled coming back out. And the analogy that occurs with glucose would be if it's in the 2 position, can go into the glucose, into glycogen, and then come back unlabeled and secreted again. So that's about a 30% difference, and yet it's going to result in a different effect on labeling. And I wonder, and there are other substrate cycles within the gluconeogenesis, glycolysis pathways as well. And I wasn't clear how you were accounting for that, but it seemed like in the 2 position, you're going to lose more in that cycle, for example, than you would in the 1 position or the others. And am I missing something, or is that part not really accounted for in your method?
Yes, I agree. There's a cycling. I think the glycolysis and gluconeogenesis can occur at the same time. They can contribute to some errors in the analysis there, but we precisely did not measure the cycling in their pathways. Yeah, but one of them shows the indirect contribution gradually actually increasing over time. I think that's related to the cycling. The cycling, because it's recycled through the system, it contributed to the gradual increase right there. But we did not measure, how much of it is actually cycled, or how much is the,
Another
the
question.
flux through
Yeah,
that one.
please.
Yes, I didn't catch this, but if you administer labeled glycerol to, say, a normal resting person, what percentage of the glucose comes directly from gluconeogenesis, what percentage has gone through other pathways?
What is the contribution of the direct from glycerol to glucose directly? What percent do you think?
That's the deuterium NMR. I answered that questions. Glycerol contribution is actually lower compared to glycogen or TCA cycle contribution. Usually it's between 10 to 20% contributions.
The direct is 10 to? Is that what you said?
Yeah. The glycerol contribution to glucose production is roughly 10 to 20%.
I see. Okay, thank you.
Thank you very much. I'll introduce. Our next talk is by Cecilia Dinitz-Spin from the University of Colorado, entitled Oral Glucose Tolerance Tests with Tracers to Different Patient Populations.
Okay, so I'd like to start by thanking the organizers for the opportunity to talk about this work. This is the first time we're talking about this, so I hope that you find it interesting. So we're focusing today on trying to understand lipolysis. And lipolysis is the breakdown of these stored triglycerides to be used, closer. All right, sorry, is that better? Yeah, okay. So lipolysis is describing the breakdown of stored triglycerides in adipose tissue. And as we know, lipolysis depends on the availability of exogenous glucose. So when you're in the fasted state, there's not a lot of exogenous glucose around. There are higher rates of lipolysis. And then in the fed state, when a lot of that exogenous glucose is available, there's insulin-mediated suppression of lipolysis. And insulin resistance of adipose tissue can make that suppression of lipolysis that's happening in the fed state either slow or incomplete. And this is a really important aspect to study because it's thought that adipose insulin resistance contributes to other insulin resistance in other tissues due to this increased circulation of free fatty acids, and therefore is very involved in the etiology of the disease. So it's known that glucose-insulin dynamics are different in adolescents, and adolescents is the patient population that we're really focusing on today. This shows some recently published data from the RISE consortium, where here the levels of glucose are the same. Am I doing this properly?
You had it for a second.
Let's press it harder.
I'm sorry.
Okay, there we go. So the levels of glucose are pretty similar here across this oral glucose tolerance test, but you see this elevated levels of C-peptide and insulin in the teens here in red compared to the adults in blue. And that high level of insulin particularly is associated both with insulin resistance that's associated with puberty, as well as this ability of these participants to produce such high levels of insulin. So you see much higher insulin responses in adolescents than you typically would in adults. And it's known that this insulin resistance is also present in the adipose tissue in adolescents. So here we're kind of bringing together some data from a couple different clamp experiments, but they show that at these different levels of the clamp, we have this nice suppression in the adults in blue, and that suppression is much slower, so we need higher doses to get suppression in youth, and we're still not kind of getting down to these low levels that you see in adults. So the goal of the work that we're focusing on today is to develop the methodology to assess adipose insulin sensitivity using data from an oral glucose tolerance test. And we know there are many advantageous aspects of the clamp, but with the oral glucose tolerance test, we have a more physiologic measure, and a lot of the disorders of lipolysis are really manifested postprandially. So it's important to be able to understand what's happening in this kind of setting. And in particular today, we're going to focus on how the dynamics of glycerol following the OGTT reflect features of insulin-mediated suppression of lipolysis. So why are we focusing on glycerol? Well, as we've already talked about today, during lipolysis, our triglyceride here is broken down where this glycerol backbone is separated from these 3 individual fatty acid chains and released into the bloodstream. But glycerol has some advantages as a marker of lipolysis compared to the free fatty acids. So first of all, as summarized here in this nice figure from Dr. Wolf's book, free fatty acids are subject to both intracellular and extracellular recycling. And so this makes it difficult when you're looking strictly at free fatty acids to know what's actually describing lipolysis. In addition, there's some work from Dr. Parks's lab showing that insulin can promote free fatty acid uptake in the periphery. So here, if we're trying to pull apart the different effects of insulin, we have sort of multiple effects of insulin, both on the suppression of lipolysis as well as promoting this uptake in the periphery if we look at free fatty acids. So we're focusing on glycerol. The participants in our study are adolescent girls, all at Tanner stage 5, with a BMI greater than the 90th percentile and sedentary activity. We excluded participants with type 2 diabetes or any medications that affect insulin sensitivity. And the data I'm presenting are all collected as part of a cross-sectional trial. So the study design here was that 1 day prior to admission, the participants had an isocaloric diet. They refrained from physical activity, and then they were admitted. We ensured that they had that overnight fast, and then they had this primed constant IV infusion of glycerol starting at 6 a.m. Then at 8 a.m., so after 2 hours, we had them ingest this 75 grams of glucola and then did regular blood sampling. And this is just a summary of the protocol showing that glycerol infusion starting at 6 a.m., then the drink at 8 a.m., and then the frequent sampling here. I'm showing it out to 6 hours. We actually started with 4 hours and then realized that we needed to extend to 6 hours. So most of the data I'm presenting will show you for the first 4 hours, since we have that for all participants. So in our tracer calculations, all of our enrichments were determined using GC-MS spec. We corrected for background enrichments, and then we used these enrichments to look at the rate of appearance using Steele's non-steady state equation, which we've already heard about a bit today. It's been used already to look at glycerol RA and clamps, and we wanted to think about it here in the dynamic setting of the OGTT. So now I'm getting to the mathematical modeling side of the talk and how we're doing this, and I wanted to start just by reminding you of some of the class of models that we're going to be talking about today, here focusing on the oral minimal model for glucose. So the oral minimal model for glucose is a one-compartment model where we think about the glucose dynamics here as this balance between a rate of appearance, where contributions are coming from the ingestion of the drink, as well as basal endogenous glucose production. And then we have disappearance here due to uptake by the liver and the periphery. And so the equations describe the glucose concentration with both the insulin-independent and insulin-dependent glucose uptake, this basal glucose release, and then the rate of appearance coming from the meal. Then the insulin action term here is given by X and is driven by our insulin concentration, which in this case is just our measured insulin concentration. And using this model, we can compute an estimate of insulin sensitivity as the ratio of these two parameters, the P3 and the P2, that are involved in the dynamics of insulin action. This idea has also been used to develop mathematical models for free fatty acids, mainly in the setting of the IVGTT. So here, Boston and colleagues described free fatty acid concentrations, remote glucose, and plasma glucose together using a delay differential equation approach. And then Parawal and colleagues described glucose, insulin, and free fatty acids using a differential equations compartmental model approach, more similar to the minimal model and what I'll be presenting today for glycerol. In their approach, they explored several different forms of an insulin-dependent rate of lipolysis. So that would be this L of X term, where X, again, is insulin action. So we have the insulin-dependent rate of lipolysis, and then just this proportional uptake of free fatty acids. So the model that we're developing for glycerol is using the same sort of ideas. We have a one-compartment model where we're describing the glycerol in the plasma here. The rate of appearance is coming from lipolysis of the adipose tissue, and then the majority of uptake we're assuming is happening by the liver. And in this original model, we're only describing glycerol concentrations, and so here the action of insulin is actually implicit, because as insulin suppression takes place, or insulin-mediated suppression takes place, that affects the RA, but we're not explicitly looking at insulin in this setting. So just to emphasize a few of the assumptions in this model, we're assuming that glycerol is in a homogeneous instantly mixing pool. We're assuming that it's representing adipose lipolysis, so we know that some glycerol may originate from other sources, but we're not accounting for that. So we're using it as our marker of lipolysis. And we're assuming this concentration-dependent glycerol uptake, so this term. We know that the majority of glycerol uptake is in the liver, but there are other tissues that may also be involved, and that could give different kinds of kinetics here than you can easily capture with a single exponential. So that's sort of a simplifying assumption of the model. Within the model, one of the important pieces here is this rate of appearance of glycerol. We assume that there's a piecewise linear function of this form, and then use this to capture that implicit insulin dependence, as I mentioned. Now, in determining what this rate of appearance should be, we have several different choices. One of these is to estimate it directly. So if we just look at the form of the model here, if we fix the volume of distribution here, then this is an identifiable model for all of our parameters, and so we can just straight estimate the rate of appearance, or we can approximate it using Steele's non-steady state equation. And then we fit our parameters to our glycerol data. So here we're just doing a constrained optimization using built-in MATLAB functions. So we've already seen Steele's non-steady state equation today, so I won't spend a lot of time talking about this. But I just want to emphasize, so when we do this in practice, we're just doing a numerical approximation of this derivative. In doing this, we get this approximation from RA for one of our representative patients. Now, there are a few known limitations for Steele's non-steady state equation for glucose, and many of those also translate to using it for glycerol. So one of the biggest ones is that the volume of the compartment changes in time. And for us, things are a little bit better for glycerol than glucose, because we can at least assume that plasma is the main compartment that we're focused on. But it also assumes that we have this instantaneously mixing pool, which isn't true for glycerol. We also know that that numerical approximation to give us the change in enrichment may be something that our Steele's equation is sensitive to. So in best practices, you try to keep that change very slow, but this is something that's changing for us. And also, as I mentioned before, we're not accounting for potential multi-exponential washout kinetics, which also affect the assumptions of Steele's equation. So to compare some of the glycerol RAs that we get using these different techniques, here in red, I'm showing you that Steele's estimate that we got. And then in blue, I'm showing you two different estimates that we're just getting from parameter fitting. On the left, we have parameter fitting where we actually fit the fasting level. So we constrained this level to be the same as what we got with steels, and then we estimated the levels throughout the rest of the RA. And for the right-hand side in green here, we're just showing the only constraint is that the parameters be non-negative. And so then with that, you can see we're actually starting at a lower RA, and then we get this kind of curve. So just to compare the differences, we see sort of a range of different possibilities using these different techniques for modeling glycerol RA. Now, when we use that RA to actually determine what the glycerol concentration looks like, we also get a couple different traces. So just using the Steele's calculation for RA, then we get this blue trace, and the red shows you the measured glycerol concentrations. And so you can see that they're recovering, and this isn't captured by the model because it didn't have that recovery in Steele's. When we use the RA that's largely estimated but is constrained at the beginning, we improve the fit, and here now we also see the recovery. And then if we just allow the best possible RA, then we get, in terms of the estimation, then we also get the best possible fit to the model. So for the rest of the talk, I'm going to be using this form of the model actually to describe different kinds of glycerol traces. So this is the participant that we've been focusing on, and here you can see that this participant has extended insulin secretion, sort of indicative of prediabetes. Here's another participant who did not recover lipolysis or their glycerol concentrations within the 4-hour time period and have this very elevated insulin response, similar or indicative of an insulin-resistant adolescent. And then here's a participant where we see there's a recovery here, but within the insulin, we can see this 2-phase insulin response suggesting that we had the insufficient initial insulin release. So from these glycerol time traces, we wanted to try to pull what features we could use to understand what's happening in terms of adipose insulin resistance. And the main questions that we wanted to look at, or the main features that we were focusing on, are how quickly does lipolysis suppress, how much does it suppress, when does it recover, and what's the impact of insulin dynamics. And so here, the metrics that we wanted to compare this to are several published measures of ways of determining adipose insulin resistance. So things looking at fasting free fatty acids times fasting insulin, fasting glycerol RA times fasting insulin, and fasting glycerol times fasting insulin, and then a whole body insulin sensitivity measure that's computed with that oral minimal model that I showed you before for glucose, here implemented, or the oral minimal model in general here implemented over 4 hours in SAM2. And so first we looked at our adipose insulin resistance measures. You would expect that if we have whole body insulin resistance, that would be potentially correlated with adipose insulin resistance. And we found that this was true for our fasting free fatty acid times fasting insulin metric, but not for our other metrics in this case. So then looking at the different features of our glycerol trace, we have how quickly does lipolysis suppress. So here we looked at the area above the curve here from the 0 to 90 minutes, so during that initial time when we would expect suppression. We also looked at the time of 90% suppression, so whenever this 90% line would hit, the 95% suppression, and the percent suppression at time t equals 50. So for all of those measures, we found that there were no associations with either the ADIPO-IR metrics or for the whole body insulin sensitivity. We did see an association between glycerol area over the curve and the average fasting glycerol. And this indicates that if we have higher rates of lipolysis initially in the fasted state, then this is somehow associated with faster and or more extreme suppression. To look at metrics of how much lipolysis suppresses, we had the maximum percent suppression for the individual and the glucose concentration at that point. Again, these measures were not associated with our measures for ADIPO-IR or whole body insulin sensitivity, but we again saw this association between the maximum absolute suppression and fasting glycerol. So again, indicating that higher rates of lipolysis initially give us, in some sense, more suppression. So then we looked at when lipolysis recovers, and to quantify this, we looked at the absolute suppression at T equals 200 and at 240, and the percent suppression at those times. And here we found that both the absolute and the relative glycerol suppression at T equals 200 were associated with the whole body insulin sensitivity. And this tells us our greater suppression is associated with a lower SI in our correlation here. So suggesting that these dynamics of glycerol recovery really relate to insulin sensitivity. And if glycerol is not recovering, then we have lower insulin sensitivity. So to conclude here, our current model accurately describes glycerol dynamics and gives us this smooth curve for glycerol concentrations over broad patterns of insulin secretion that we observe across individuals. Our preliminary results suggest that it's really the dynamics of recovery for glycerol that are most strongly associated with a whole body glucose insulin sensitivity. And future work here will focus on explicitly including insulin in these models, and ideally eventually getting to the point where we have, from our explicit insulin representation, a way of estimating adipose insulin sensitivity analogous to the whole body insulin sensitivity. So I would like to acknowledge my collaborators on this work. So my student, Kate Bubar, is a very talented undergraduate who is involved in a lot of this analysis. My PhD student, Kai Bartlett, has done all of our glucose insulin sensitivity measures. And then my collaborators, Dr. Cree-Green, and our other collaborators at University of Colorado, Denver, and Anschutz. Thank you.
Bob Hickner, Florida State. It seems your point about the high initial glycerol concentrations being related to the greater suppression, but that would probably relate or depend on who you're studying at the time, because if you have obese populations or well-trained populations, you might see something different than a sedentary lean or lean otherwise group. Can you comment on that?
Sure. Yeah, I think some of these things could definitely be different across populations. And as I said here, we're focusing on obese adolescents. I think getting a better perspective on that could also come in if we have included the insulin, so that we have the insulin explicitly present in the model.
Yeah, on another point, I appreciated your statement that the glycerol could be taken up into muscle and other tissues, and the amount of that uptake and the relation to the VLDL, for instance, and other modes of action of getting, having glycerol in or out. You would have a situation where during, say, hyperinsulinemia, that factor might change differently in different populations, so following food intake. So there are a lot of possibilities for potential error in the model based on the relative contributions of where the glycerol is going, other than just the hepatic removal of glycerol.
Right, yeah, I totally agree, and I think that really shows us, those kinds of aspects of the physiology are things that really make it problematic to try to, say, use Steele's non-steady state, and that's why using a more flexible form can actually address that better, because it's not making a specific assumption of that. But again, to fully account for that, we may need to bring in multi-compartmental approaches, other kinds of of complexity to the modeling approach. Mhm.
There's not, I have a third one. It's that no one brought up or no one instead of, and asked about this little blip in the beginning. In the first 50 minutes, you have a biphasic almost in the steel model. Can you comment on why that little blip is there, you think?
Yeah, so that was just for one individual. So, this wasn't something that we consistently saw over everybody, but I think, again, we know that there are some of these limitations associated with steel, and some of them people have addressed in different ways. So one of the ways is to sort of downsample and actually estimate your RA with fewer time points. And so that's one thing that you could possibly do that would sort of smooth out some of the variability that you see in the RA there.
Yeah, I'm caution smoothing out if it might be something real. But, yeah.
Right, it's always a balance.
Because these girls are obese, and they may have some delayed gastric emptying. It could be delayed gastric emptying in that individual. She showed the insulin trace, and they had a small peak in insulin, came back down, and then went up to a large hump. And that's why, that's why it may be that this model really needs the explicit insulin rather than the implicit insulin, and that would then explain those bumps.
I'm not sure where the glycerol is taken up makes a difference. Because you're modeling RA. So what difference does it make where it's taken up?
So the place that it's coming in terms of where it's being taken up, is in the assumption about the glucose decay, or the glycerol decay. So there, if we assume that it's just this single exponential, then that's assuming either one main place that it's being taken up or similar kinetics in the way that it's being taken up across the board. And there's a compensation between the SG and the RA and in actually describing glycerol concentrations. So I think it impacts what happens with your RA, not so for the overall model, it's impacting what's happening in RA because you have that potential interaction between parameters.
Okay. Thank you very much.
This concludes our session. If you did get an evaluation form, we'd love it if you filled it out and turned it in. Thank you.